Okay. Melon is an oily seed that is grown widely in Africa. It is cultivated for its relatively high percentage of protein and fat. It also contains essential minerals like calcium, magnesium, and essentially the oil content of it is also very rich. So in terms of processing, melon is processed through depuding, fermentation, washing. After washing, we have shelling, and shelling could be done both manually and mechanically. We have several products from melon, like robo in Yoruba land. Melon could also be prepared as a thickener for soup and some other products. In terms of storage, melon could be stored in grinded or ungrinded form. And one of the greatest ordeal of melon seed is that during storage, it deteriorates due to microbial attack. And most of these microorganisms are being introduced during the processing steps. These are the justification. Shelling, either mechanical or manual, being the last stage in melon processing, is the last stage prior to consumption of melon, and is barely the most critical stage in the processing. And because it's the stage that is last before melon is being consumed on the table and at home. So some of these microorganisms could be introduced through the process of shelling, just as I've said. Therefore, in light of this, there is need to give the consumer a guidance so that they could be able to purchase wholesome product in the market. So the objective is to compare both the microbial status and the nutritional composition of both handshell and machine shell melon seed. Materials and method: 1 kg of handshell of both handshell and machine shell melon seed were purchased from Omida Market in Abeokuta. These melon seed were transferred aseptically at 4 degrees centigrade and kept at 4 degrees centigrade prior to the analysis, that is microbial, proximate, and chemical analysis. For the microbiological analysis, the melon seed was blended, and we do analysis such as total bacterial count, staphylococcal count, coliform count, and fungal count. Also, it is instructive to know that at the end of the microbiological analysis, 2 isolates were picked, 1 each for both handshell and machine shell melon seed for molecular analysis, for molecular identification. The proximate composition and the chemical composition were determined using a well-known analytical procedure. Like for the chemical composition, essentially we determine both peroxide value and free fatty acid, because we want to know whether about the state of rancidity that is going on in the melon seed. Data were analyzed using SPSS version 16. These are the results from table 1. Table 1 shows the mean proximate and chemical composition of both melon samples. It is shown here that all the parameters identified there's no significant difference between those parameters in both handshell and machine shell melon seed, except the pH. And we think that the pH is the difference in pH could be as a result of different level of biochemical and microbiological reaction that is going on within. And it's also noteworthy to say that both peroxide value and free fatty acid value obtained for both melon sample seeds are within, they are even below the Codex Alimentarius value of 10. So the implication of this is that the oil that could be expressed from that melon, both melon seed, is edible. And the findings of both proximate and chemical composition of those melon samples, the findings are within the range reported by previous researchers like Oluwa Bamewo et al. 2015, Ogundele et al. 2012, and many other researchers. So this chart is revealing the microbiological analysis result. First, we have the total fungal count. In this chart, the total fungal count for ant shell melon seed is, for machine shell melon seed, is significantly higher, was significantly higher than that one of ant shell melon seed. That could be as a result of the remnant of melon shell in the shelling machine, in the sheller that was used to shell the melon seed. And also it could be due to fungal spores that is already landed, that has landed on those shelling machines. Also, furthermore, we see that the total bacteria count for both ant shell and machine shell melon seeds, there's no significant difference. For the third chart, we can see that the total staphylococcal count is signified with TSC. It's obvious that the total staphylococcal count of ant shell melon seed is significantly higher than that one of machine shell melon seed. I think this is expected because Staphylococcus, as found out by many researchers, and as we all know, they are ubiquitous, and also they are normal microflora on the skin of human beings. Possibly, they could have gotten into the ant shell melon seed during the shelling operation. For the total coliform count, the value is very low for both melon samples, and the low value obtained here could be as a result of nutritional competition between the microorganisms in both samples. These are the biochemical results for all the microorganisms in both melon seed sample. And this one shows, the chart here shows the percentage composition of the earlier found bacteria. We can see that the Staphylococcus aureus and Bacillus subtilis are both common to both ant shell and machine shell melon seed. The two organisms, they are common. It could be as a result of the fact that they are very, very common in the environment. Just like I've said, the Staphylococcus aureus is very common as a normal microflora on the skin of human beings, while Bacillus subtilis is also very common in the environment as a source of the presence of the spores of the Bacillus subtilis in the environment. It could have possibly come in contact with some of those melon seed samples. Other bacteria were also obtained. Now, this one shows, the chart here shows the composition, percentage composition of the fungal species found in both melon samples. But it is instructive to know that the commonest are the Aspergillus species. This one shows, as I've said earlier on, that we picked a sample each, an isolate each, from both ant shell and machine shell seed for molecular evaluation. So from here, both samples, sample one, which is Aspergillus niger, we did comprehensive microscopic and microscopic evaluation for both the two isolate picked. And from there, we proceed to molecular evaluation of both isolates This is the gel picture of the extracted genomic DNA. And this is the PCR amplicon for both samples. So at the end, we were able to confirm that isolate 1 was found to be 100% Aspergillus niger strain, while isolate 2 was found to be 98% A. flavus strain. Conclusion: based on the findings, we can conclude that both melon seed, that is hand shell and machine shell, being sold in the market, in an Abelvita market, contain acceptable level of nutrient beneficial to man. As we can see that all the proximate composition and the oil is also non-rancid. We can see it as I presented earlier. 2, but regardless of the fact that it contains acceptable level of nutrients, both harbor microorganisms. But the machine shell contain more microorganisms, especially mold, than hand shell melon seed. And therefore, you could make it to spoil faster than hand shell melon seed. Based on the conclusion and the findings, we put up forward 2 recommendations. 1, melon processor and sellers are advised to cultivate and adhere to good handling practice in the course of their routines. 2, melon seller in the market are advised to keep melon in a covered container prior to sale to protect the seed from mold spore and bacterial contaminants in the environment. Thank you for listening.
All right. So thank you very much for your talk. And I think in the interest of time, we might just ask people to to write their questions, and we will take the question and answer session for all the speakers together. Okay, so thank you very much, Mr. Shotaya, for that talk. If you can unshare your screen, and let's go on to the next person. I just want to also note that each talk should be as brief as possible, so that we can stay within the time we have. Thank you very much. So our next speaker, could we have you? So Dr. Sholale would be giving us his presentation. Over to you, sir.
All right, thank you very much. Can you hear me?
Yes, we can.
Yes, we can.
And you can see my screen, right?
Yes, too.
All right, so thank you very much, and I welcome everyone to this presentation. I'll be sharing on the research work we've been doing for a while. And my title for this morning presentation is Global Microbiome Study, discovers thousands of new species, maps urban antimicrobial resistance, and reveal new drug resistance. And so the journey for this project started with trying to understand the interaction between humans and the built environment. And it was this that gave back to the meta-sub project, which is metagenomics and the meta-design of the subways and urban biome. It started, as consortium in 2015, and as far as 2020, we've been collecting samples, and we have these major aims for this study, that is to create geospatial metagenomics and forensic genetic maps to identify and track antimicrobial resistance in the urban built environment, and to identify novel biosynthetic gene clusters for drug discovery. And so what happens is that every June 21st, we hold what we call the Global City Sampling Day, where we have participating cities around the world. We go out with their volunteers for what we call a swap venture. And our participants, are trained on how to collect samples, how to capture metadata using their mobile phones. And each participant or pairs of participants are then required to, start collecting, samples using their mobile phone, like I earlier mentioned. So our samples are usually collected using a synthetic swab, which you could see on this screen with wearables. You could see the swab stick, and you could see someone, taking samples. And so what we do is we visit train stations, cities around, or participating cities visit their train stations and to collect samples. And we also visit our own, train station here in Nigeria to, collect samples. And once this is done, the samples are sent, are sent to the lab, which are then also stored in a freezer till, they are ready for DNA extraction, library preparation, and sequencing. Many of our city sampling days wouldn't have been possible without members of each cities, which we usually call the swab squad. As you can see, this group of volunteers who participated in Nigerian study. And they usually include NYC core members, students, and even volunteers who are graduates too, who are interested in what we are doing. And like I said, we have about 60 cities currently, across the continent participating in this study, which also include Nigeria. And our focus is at Ilorin and Offa, where we have train stations where we go there to collect samples. At Ilorin too, we also have the bus transport station too, where people take buses, but for sort of time now, they've been having problems, but then we've not been able to sample in the last 1 year with them. So these are the sampling sites. And so between 2015 and 2017, we've collected nearly 5,000 samples across, the 60 cities in about 32 countries and also covering about 6 continents. And the designated lab, analyze the samples after they've received our samples using a genomic sequencing technique called shotgun sequencing to detect the presence of various microbes, including bacteria, archaea, that is single-cell organisms that are distinct from bacteria, and also viruses that uses DNA as their genetic material. And 2 months ago, we have this publication that came out of the work that we have done so far. And so just to quickly now give us some of the highlights of the study, we discovered that there is a consistent core urban microbiome signature, found across the data, which is distinct from organisms commonly found in the human microbiome. And so in essence, what we are saying is that we could see difference in the type of microorganisms that you see in the built environment as against what can be found on human. And for this, we feel that there is need for further research on building materials and its influence on the microbiome that could provide insight to civil engineers, architects, and city planner, just to ensure that we have a safe city. And another thing was that there could be factors and features that can influence a city microbiome, which could be the population of that place and the population density. I also want to talk in terms of the elevation of that place. There may be proximity to the ocean and the climate. And so the finding about this distinct feature could enable future forensic studies to know how cities or built environments acquire, their microbiome. And so from this study, some of the cities have discovered many novel species and molecules in which we are able to identify about 10,000 plus viruses, about 700 plus bacteria that were discovered across data sets that were not present in any reference database. And furthermore, we also discovered that there are small molecules and proteins that are made by microbes that could also lead to discovery of new antibiotics and other, drugs. Discoveries made about new microbial species will also lead to new laboratory tools and approaches, such as a novel way of using molecular editing tool known as the CRISPR. In this study, we were able to also identify about 800,000 plus of CRISPR arrays, which are snippets of viral DNAs that were found in bacteria, and about 4.3 million plus of a new peptide, that is small proteins, were also identified from our studies. Going on, here is talking about antibiotic resistant genes. We discovered that antibiotic resistant genes are widespread in cities, and predicting antibiotic resistance from genetic sequencing, we know that could be quite challenging. But based on this work, we were able to map some genes that are known to be linked to resistance, and we're also able to quantify their abundance and also confirm their genetic, markers. And we also find that some cities, add more resistant genes than others, and there might be cities with specific signature for some of these genes. And for this too, there is still need for more further studies to fully grasp the scope of the clinical impact or clinical relevance of this kind of result that we have. And so going forward, so going forward into the future, you know that we have something in town now that, the coronavirus. And so the coronavirus pandemic has brought a new interest in the project, as we hoped that we'll continue, sampling the biome of the environment. And this year, 2021, too, the global city sampling, is done, and and we are still maintaining, the major aim of our study. And so, just for you to see some of the outcome of our study, I'll be just giving a short demo. And that is one of our first aim of this study, that is to create a geospatial metagenomic and forensic genetic map. And I just want to quickly state that at this point, our collaborators are welcome, to look into Nigeria data as we seek to identify good bioinformaticians and scientists who can ask new questions based on the available data that we have in Nigeria. So I want to thank you for listening to this presentation, and you can as well follow our research. And so quickly, I will just switch. I believe you could still see my screen. And so this is one of the outputs of our work, and this is a metagenomic map of the city. And I will just scroll zoom to Nigeria, where you could see some of the data that we've collected over the years. I want to encourage you guys to visit this site and just play around, and you can see that you'll be able to harvest information in terms of the abundance of microorganisms that are identified based on our study. You could get information about the metadata about the organism that you pick from, let's say, this sunburst view. Quite a lot of interesting things that you'll be able to pick. And I'm happy with this kind of study, that in the sense that from Nigeria, we're able to identify that there are unique bacteria that came out from this study. So I want to thank you all for listening to this presentation. God bless you all. Thank you. Hello.
Thank you very much, Dr. Sholale, for the presentation and doing it in a timely manner. So could we have the next speaker? Also, definitely, I would reiterate the fact that if there are questions, please, the chat box is still there. Speakers and the audience can interact there while we wait for the overall question and answer session. So over to the next speaker, please. Thank you.
Is that me, Christian?
Yes, Christian.
Okay. A moment, let me share my screen. Can you all see my screen?
Yes, we can see it, Christian. Go ahead.
Okay, thank you very much, Dr. Yinka. Good afternoon. My name is Christian Anumodu. I'm a postgraduate researcher in the School of Chemical Engineering, University of Birmingham. Today I'll be talking about the production and optimization of bacteriocins from lactic acid bacteria. I'm going to start with a brief overview. Food safety is everyone's business and is really important with regards to public health. So microorganisms have a way of causing problems in food, and currently there's a lot of challenge associated with their presence in food, which is mainly food spoilage and foodborne illnesses. The WHO estimates that every year we have about 600 million cases of foodborne illness with around 420,000 deaths. And importantly, more than 30% of this is happening in children below the age of 5. So it's imperative that food should be preserved or processed in such a way that microbial factors will not lead to diseases. So a variety of approaches are currently being employed in food preservation. You use chemical preservatives such as your benzoate or your sorbates. However, there's challenges associated with the use of these chemicals. The first one is consumer perception of their safety. And also they have a way of altering the organoleptic quality of foods. So physical methods such as high temperature or low temperature also have their own problems. Like high temperature may lead to the formation of heterocyclic aromatic amines, which are said to be carcinogenic and other things. But so our research in the University of Birmingham School of Chemical Engineering is focused on biopreservation and specifically the use of bacteriocins from lactic acid bacteria. Bacteriocins are ribosomally synthesized during the primary growth as a primary metabolite during the logarithmic growth of the organism. So they are not antibiotics, but they have antimicrobial effects. A variety of bacteriocins have been produced, including niacin, which has attained the status of being generally regarded as safe. Their use in food is encouraged because they are natural, being synthesized from organisms, and they have high stability, pH stability, temperature stability, osmotic stability. So food products that are fortified with these bacteriocins have a way of remaining safe as the antimicrobial peptide remains active throughout the process in which a food receives. Also importantly, because of the rise in antimicrobial resistance, we are very cautious about what goes into our body. Importantly, because they are peptide in nature, they are broken down by peptic acids in the stomach, so they do not get into systemic circulation. The aim of our research is to screen and identify bacteriocin-producing lactic acid bacteria from the dairy products, cheese, milk, yogurt, etc., and to optimize the production of bacteriocins by supplementing their growth condition. And we are in this my research, we used whey, which is a byproduct of cheese production. It is very cheap, and we supplemented it with different sugars and amino acids to monitor the production of bacteriocin. However, with regards to the bacteriocins, there is a challenge which is the bacteriocins do not remain active in food over a long period of time because they sometimes can be inactivated by the food matrix. And after a short period of storage, they are no more effective in inhibiting microbial growth. So to solve that problem, we are looking at the microencapsulation of bacteriocins using chitosan and sodium tripolyphosphate. This is the methodology we used. My primary isolation of bacteriocin-producing lactic acid bacteria was achieved using the Mano rugosa and shaped agar, which is selective. And preliminary identification was by the API kit by Biomerius, which incorporates 48 tests, which helps me to simultaneously run the analysis and identify the possible organism. Of course, we are going to go forward later on in the research to do molecular analysis of the organism to identify it by cysteine RNA sequencing. So produced bacteriocins are concentrated using ammonium sulfate precipitation method because of the peptide nature, and we evaluated their antimicrobial activity against Bacillus cereus and Bacillus subtilis. And we chose these two organisms because they are important with regards to food spoilage, as they are spore formers and can survive some of the treatments which food received. And of course, we went forward to optimization, enzymatic characterization, and the micro encapsulation of the bacteriocins. So we were able to isolate two lactic acid bacteria, Lactococcus lactis and Lactobacillus acidophilus, and we made the identification using the API kit, as can be seen here. So we got good protein yield at 54 milligram per liter, and also a good total bacteriocin activity against the test organisms. However, we went forward to do a co-culture assay because sometimes your test organism behaves differently in, will I say, an in vivo environment as against an in vitro environment. So we used the Costa plate to co-culture our Lactobacillus and Lactococcus with the test Bacillus species. And in the chart on my right-hand side, you can see that the growth of Bacillus cereus and Bacillus subtilis was significantly inhibited when they are co-cultured with Lactococcus lactis and Lactobacillus acidophilus. So we got an MIC value of 50 microgram per mole for Lactococcus lactis and 100 for Lactobacillus acidophilus. The value we got for Lactococcus lactis is quite nice because it's comparable to what is gotten in literature. So for the optimization of the growth of, now we are working with only Lactococcus lactis because we got the best MIC value from this particular organism. We used a Plackett-Bowman experimental design using 5 different variables: temperature, pH, sucrose, inulin, and tritone at 3 different levels, as can be seen on the chart on my right-hand side. And the produced bacteriocins was used to inhibit the test organisms, Bacillus cereus and Bacillus subtilis. On my right-hand side, you can see the chart. The first one is the effect of different combinations on the growth of the organism. And below, we have the bacteriocin production. So from the chart, it is easy to note that the run number 9, which we used sucrose at a concentration of 30 milligram per ml, tritone at 10 milligram per ml, culturing at 30 degrees centigrade, gave us the best growth of Lactococcus lactis and also bacteriocin production. So going forward in all the research we are doing, we are using these particular parameters to culture for our organism. Next, we did microencapsulation studies using chitosan and sodium tripolyphosphate, cross-linking both of them with our bacteriocin. We've not identified the bacteriocins, the specific bacteriocin anyway, cross-linking it to get ionically charged nanoparticle. The nanoparticle we obtained, we did a dynamic light scattering DLS analysis to determine the size intensity. So on my right-hand side, you can see the first chart, we have pure niacin commercially obtained from Sigma Aldrich, and we did a DLS analysis of the chitosan. And finally, we can see a DLS chart of the microencapsulated bacteriocin. So the microencapsulated bacteriocin had a size of 146 nanometer, and it was monodispersed, which is okay. And we had an encapsulation efficiency. It is important to note the efficiency, because if the encapsulation is not efficient enough, we'll end up with very low quantity of niacin being encapsulated. And we got an encapsulation efficiency of 60%, which is okay. And a zeta potential of 30 millivolts. The zeta potential gives an indication of the stability of the nanoparticle. The next thing we did was to inhibit the test organisms using both the encapsulated encapsulated bacteriocin and pure niacin that we obtained commercially. And on this chart, we can see the effect or its effect on the growth of Bacillus cereus and of course Bacillus subtilis. So looking at the gray chart, which is our encapsulated bacteriocin, we found that because we had a 60% encapsulation efficiency, their ability to inhibit the test organisms was a bit limited compared to niacin, which is the orange, which has the orange line. However, if you notice, because this fortification was done over a period of 5 days, if you notice from the chart, although there is a rise in the growth of the test organism, after 60 hours, the rate at which our encapsulated niacin inhibited the test organism was better off than the free niacin and that of the cell-free extract. This points to the fact that the encapsulated niacin is more stable over a longer period of storage compared to the free niacin, which may have been broken down by the food matrix. And after 5 days, the encapsulated niacin was able to reduce the microbial count in our medium compared to that of free niacin. Although this points to a success in our research in extending shelf life using microencapsulation, but because of the rapid growth of the test organism after 24 to 48 hours, we can't say we've arrived at where we are going, because this is the point where spoilage or possible the production of azotoxins by organisms in food happens. So further research we will be doing is to find a way to make the encapsulated bacteriocin quite effective at a shorter period of time. So in conclusion, this research is still ongoing and we've not gotten to El Dorado yet. However, we've been able to obtain bacteriocin producing lactic acid bacteria from prime cheese, and we've synthesized the bacteriocin using very cheap substrates at a very high efficiency. So future work will be directed towards the molecular characterization of the isolated organisms, and also maybe HPLC purification of the bacteriocin we've synthesized to actually determine if it is a new bacteriocin, because just like the antibiotics, the quest for newer bacteriocins with better activity is ever going. And of course, we are going to try and employ the Huddle concept using other approaches to combine with our bacteriocin to see if we can reduce the growth and proliferation of pathogenic organisms in food. And we are thinking about the use of different essential oils, especially for the inhibition of spore outgrowth, because from this research, the niacin and the encapsulated bacteriocin we have was able to inhibit the vegetative forms of the organism, but not the spore form, which was able to grow once the inhibitory factor is reduced. So thank you very much. And if anyone has any questions, we can, I'm ready to answer it. And I want to especially thank my supervisor in the person of Dr. Helen Oyaka, who happens to also be a member of NAM, for bringing me in, for supervising me, and for all the help she has rendered. Thank you very much for this opportunity.
Thank you very much, Christian. And yeah, we appreciate the very succinct